Welcome, everybody, to the Ultimate Health Model podcast, where we take a deep dive into uncharted waters on everything health. Today, I have the distinct pleasure and honor of welcoming Professor Thomas Seyfried, who is a cancer researcher at Boston College. He's been doing this for 20 years, is more than that, a long time. So anyway, so we're really excited to have him. I want to tell everybody, this is a very important podcast. We all know people, our friends listening, we all know people who had this cancer. I've lost family members. I've lost friends, friends of family. Everyone knows somebody. And this is a super important podcast. So whoever's listening, tell your friends to listen, get around this podcast to listen to it, because this is extremely important. We all have somebody who's affected by this. So anyway, without further ado, Thomas, why don't you introduce yourself and how you started doing this and what got you into cancer research, how long you've been doing this, and we'd love to hear your story.
Well, thank you, Benjamin. It's nice to be here on your podcast. Well, I've been doing this for a long time at Yale University before I came to Boston College in 1985. We've been working a lot on epilepsy, biochemistry. I'm mapping epilepsy genes. Most of the work on cancer, though, was done not as a strategy for managing and preventing cancer, but more or less looking at the biochemical changes in lipids that occur in tumors that are already developed, and comparing abnormalities in lipid biochemistry with that of the normal tissue from which the tumor arose. So, and pretty much, characterizing these kinds of things, but it wasn't in any strategy to come up with a therapeutic strategy at all. We did that primarily from the work on epilepsy. So the work on epilepsy brought us to understand how to manage epileptic seizures using diet, ketogenic diet. But it really worked. It really was linked to lowering glucose and elevating ketones. What was the biochemical changes in the brain of an epileptic mouse or person that was preventing epileptic seizures? And we did a lot of studies, and we found it was lowering glucose and elevating ketone bodies. And the mechanism by which that diet therapy for epilepsy, which is currently used throughout clinics throughout the world, is still under intense investigation. And it's not clear yet why lowering glucose and elevating ketones inhibits epileptic seizures. But we were running models of cancer at the same time, and it became clear that it was some sort of calorie restriction that was working to stop the seizures. And then we did some work on tumor angiogenesis, which is blood vessels in the tumor. And then we found out that calorie restriction could lower the blood vessels and manage the tumor. And it wasn't clear to us what the mechanism was by which that was happening. But then we stumbled across the work of Otto Warburg, who clearly said that cancer cells need sugar, glucose, and if that's driving their dysregulated growth, a fermentation metabolism, then targeting glucose could be an effective way to manage cancer. And a person, Linda Nebling at Case Western Reserve University in Cleveland, showed that little kids with brain tumors were responding remarkably well to restricted ketogenic diets. And she said this supports Warburg's theory. So I went back and I started looking at who Warburg was. We always heard of Warburg. We just never knew his fascinating history and story. And then I began to really read detailed his work. And it became clear that he said all cancers initiate from chronic damage to mitochondria, leading to a compensatory fermentation dependent on glucose as the primary fuel. So our work on epilepsy and cancer seemed to support what Warburg was saying. Then we evaluated mitochondria in cancer, lipids, and we found that all cancers have defects in cardiolipin, a major lipid on the inner membrane of the mitochondria, regulating oxidative phosphorylation. So that supported exactly what Warburg was saying, no question about it. And then I teamed up with guys that look at electron microscopy of cancer, and we were able to show that mitochondria are damaged in structure. Just look at them under the microscope and you see abnormalities, which then supported further Warburg. Warburg never looked at mitochondria. The electron microscopy work was not developed in his time. He based his whole theory on biochemical measurements of oxygen consumption and lactic acid production.
Now you said, hold on, I just want one second while we're going so I can make sure that our list friends listening understand. The mitochondria is dysfunctional in some way in cancer cells. And I wrote from in one of your papers I was reading, if this is correct, that the mitochondrial dysfunction mutations have been reported in all cancer types?
No, not mutations. It's the mitochondria structure itself. Mitochondria has a very intimate kind of structure. It has an outer membrane and an inner membrane, and the inner membrane is extended to form cristae, and the cristae have the proteins and the lipids of the electron transport chain that allow our cells to generate energy using oxygen as a common acceptor of electrons. But when you look under the microscope, the electron microscope, and you look at mitochondria, you see vacuoles and holes and broken cristae, and you see all this kind of stuff. So, structure in evolutionary biology, we know structure determines function. Mm-hmm. So if the structure of the organelle is abnormal, the function of the organelle is going to be abnormal, which then supported Warburg's biochemical findings. But we went further than Warburg. We actually found out why there was such a controversy associated with his work. He misunderstood oxygen consumption as a marker for oxidative phosphorylation. Like today, most people still do this. We have evidence that this is not an accurate marker. He also used lactic acid as an accurate marker for energy production through fermentation in the cytoplasm, and we now know that that's not an accurate marker from our research. And he knew nothing about the fermentation of glutamine in the mitochondria. So there was a lot of confusion. Bottom line, Warburg was absolutely correct. All major cancers arise from chronic damage to oxidative phosphorylation, coupled with a protracted upregulation of what we call substrate level phosphorylation, which is in the cytoplasm through the Embden-Meyerhoff-Pars pathway, or in the TCA cycle at the succinyl-CoA ligase reaction. So he didn't know a lot he knew about the cytoplasm because he did a lot of the work on that originally, but he himself did not know that the mitochondria produce a second form of energy through a fermentation metabolism.
Oh, he didn't know that?
He did not know that because the evidence wasn't developed at that time. I was the first one to predict that, but it was pure speculation because I figured that why would these tumor cells use so much glutamine? And people say, it's respired. And I said, can't be respired because the structure is abnormal. So there's something else going on here. So then I teamed up with Christos Xenopoulos from Semmelweis University in Budapest, Hungary. And he's the world expert on that particular biochemical reaction. And I asked him to help us out here. And he started looking into it in his lab, and he was able to confirm that mitochondrial substrate level phosphorylation, which is independent of oxidative phosphorylation, could be producing energy in mitochondria that do not produce enough energy through oxidative phosphorylation. I know this is a little complicated, but unfortunately, this is the way it is. So then we launched a series of detailed experiments to test the hypothesis that cancer cells are generating energy in the mitochondria from a mechanism independent of oxidative phosphorylation. So we used labeled glutamine as the substrate.
Fermentation, you said fermentation.
Fermentation. The cancer cell can get energy from glutamine in the absence of oxygen. So that's a fermentation mechanism. So most cells in our body can't survive without oxygen. You have to realize cyanide kills humans. We can kill, if we want to commit suicide, we can drink cyanide. It kills us real fast, doesn't kill tumor cells. Tumor cells don't die from cyanide. It's because they're fermenting. The energy from fermentation doesn't require oxygen.
Now,
So cancer cells can live in cyanide.
let me ask a question. So with cancer cells, for our friends listening, so cancer cells still use oxygen, just not as much oxygen. Is that correct?
Yes, that's correct. So they use oxygen, they consume oxygen, but most of it is not for energy production, ATP. Some of it is.
Okay.
Yes, depending on how damaged the mitochondria are.
Very complex.
But a lot of the oxygen that comes into a tumor cell mitochondria produces ROS, reactive oxygen species, which are carcinogenic and mutagenic.
Which causes inflammation.
Inflammation, carcinogenic. Well, inflammation is coming from the waste products of the fermentation metabolism.
So
which
it's
is
a circle.
acid.
Yeah, so it's a circle. Basically, you get inflammation, you get more ROS, you get more damage to the mitochondria. So it's kind of a vicious circle.
The inflammation is coming from the waste products of the tumor cell. Okay, so the waste products of the tumor cell are lactic acid and succinic acid, which creates an acidic environment, leading to an invasion of immune cells, which then link to the inflammation. So it's like an unhealed. When you get a wound on your hand or leg or wherever, it becomes inflamed. And that inflammation is coming from an attack of our immune system on local bacteria, leading to a lot of cytokines, inflammatory cytokine release. Well, cancer is like an unhealed wound in our cells and our tissues. And the immune cells come in to try to put out or heal the wound. And in the process, they create a lot of inflammation. Ultimately, the ROS, the damage from the reactive oxygen species, leads to further damage to the ability of the organelle to produce energy, and also links to the mutations that you see in the nucleus. So the mutations that people seem to be focusing on are largely downstream epiphenomenon, largely irrelevant.
From the mitochondria.
Yeah. But mitochondria are linked to a lot of the mutations that we see in the nucleus, which means that they're not causative, they're effects of the.
No, that makes sense. Let me ask you a question then, and this for our friends listening, this is important. The mitochondria does a lot more than produce energy. One of the things it does, and I write this in my book, is that it controls cell apoptosis. Right. So if that mechanism or those receptors are damaged, well, that would possibly make a cell be autonomous and proliferate uncontrollably. Is that correct, Thomas?
Well, when you switch from oxidative phosphorylation to substrate level phosphorylation or a fermentation mechanism, they lose their ability. The apoptotic signaling cascade, the release of cytochrome C, doesn't kill the cell anymore. The defect in the mitochondria, mitochondria control the destiny of our cells. Everything, in fact, our whole body revolves around the health and vitality of the mitochondria. We ultimately die from old age because the mitochondria just go up and die, and you're dead. So, but in apoptotic signaling cascade, where the mitochondria control the destiny of the cell, if the cell is injured or structurally damaged in some way, the apoptotic signaling works. The mitochondria release cytochrome C, and the cell dies through a programmed cell death mechanism. So that's called apoptosis, programmed cell death. But in that chronic transition from oxidative phosphorylation to substrate level phosphorylation, which we call fermentation, that apoptotic signaling cascade is no longer working. So it's the kill switch is broken.
Yeah.
So you can release cytochrome C and do all this stuff, but the cell doesn't die any longer because it's now living off an energy source that's independent of mitochondrial. Mostly, not all, not completely, but certainly the apoptotic signaling no longer works.
Okay. So that's very interesting because, like I said, people don't know. For us, I assume a lot of our friends listening don't know that the cell or that the fate of the cell is controlled mostly by the mitochondria, and as far as you're saying, apoptosis.
The mitochondria also control the cell cycle and the differentiated state. So what I mean
It
by
controls a
cells,
lot. Yeah, it controls a
so
lot of the
what
cell's
what
functions.
that essentially means is that through wear and tear, we produce new cells all the time. So cells wear out and die. Our immune system picks up the corpses, and gets rid of them. And then new cells are produced in a regulated way. They're not dysregulated. The definition of cancer is cell division out of control or dysregulated cell growth. So who regulates cell growth? How does our body regulate cell growth? And it's the mitochondria within the cells regulate the growth and destiny of the cell. So they control the cell cycle. So when a new cell needs to be produced, the cell divides through a mitotic process, but the mitochondria are functional during the mitotic process. So they keep the number of cells regulated. We know this from liver regeneration and this kind of stuff. We know the mitochondria regulate growth. So if the very organelle needed to regulate the growth and destiny of the cell becomes corrupted, the cell falls back on ancient pathways of fermentation, which is dysregulated.
Makes sense.
All cells on our planet 2.5 billion years ago were unbridled proliferation. They just grew and grew and grew
That's
through a
interesting.
fermentation mechanism because there was no oxygen in the atmosphere at
Interesting.
that time.
Okay.
So what happens is these cells, and we have the heirloom pathways in all of our cells. So these are small, like the glycolytic pathway and the glutaminolysis pathway. These are all there, but they're only upregulated under very unusual conditions. Like, say, for example, a heart attack. A person has a heart attack. They fall down. Of course, they're not breathing. And if they don't get oxygen, you don't bring that heart pumping blood back again, the person's dead. And we all know that, right? There's not a mystery. People die from heart attacks. Why? Because their energy system shuts down. There's no longer oxygen coming into the cells.
Wow.
And what happens is the most of the energy in our cells is driven by membrane pumps, and you've got to keep those pumps with energy, otherwise the cell dies. And usually from a heart attack, that you die from brain neurons dying. So when your neurons are all dead, you're dead. But it's very interesting. When you see, if you measure the blood, people have done this on diving, long exercises, and heart attacks. If you look at the blood of patients, you see a massive amount of lactic acid and a large amount of succinic acid in the bloodstream of a heart attack person. And that's because in order to keep the cells alive for about 5 minutes, you have to switch to an immediate fermentation metabolic. There's no oxygen.
Oh.
Okay. So you see the waste products of
Instantly
the fermentation immediately accumulating in your bloodstream. Okay.
Wow.
Now, if you don't get oxygen back in there, that person is dead.
That's fascinating.
So now listen, the cancer cells are throwing out those same two fuels all the time, even in the presence of oxygen. So
makes sense. So what they're doing, let me, for our friends listening, I that was a really fascinating thing what you said, Thomas. I really like that. And for our friends listening, what we said, they go back to a primitive way of living, the cells, cancer cells, because that's what like I said, a billion years ago, that's all they would do is proliferate. So that's their only job at any cost is to proliferate.
Yes, and as Albert St. Georgi said, they proliferate until the fermentable fuels in their environment dissipate, and then they die. So they would grow and grow and grow and grow, and then divide and grow, and then based on what? Where's the fuel? Well, there's no oxygen in the atmosphere 2.5 billion years ago. There was no oxygen. That came from photosynthetic bacteria. So these cells, as long as they had fermentable fuels that could provide energy in the absence of oxygen, they would live and grow. And when the fermentable fuels were used up, they would die.
Wow.
So cancer cells are driven by the availability of fermentable fuels in the environment, which is glucose and glutamine.
And how do this is a really kind of important question. This is common, makes just sense, Thomas.
No, it doesn't.
It doesn't make sense to clinicians because when I'm talking about how glucose makes sense to me.
Yeah, well, it doesn't make sense to someone which is the majority of the cancer field that thinks cancer is a genetic disease. So what I'm saying, you might as well be talking in some language that nobody can understand.
It's called, makes sense to me. Yeah.
There's two things that you need to know about current cancer problem. Number one is evolutionary biology. And you'd be shocked to find out how few people really understand evolutionary biology.
Oh my God.
So number one, you have a lack of scientific literacy. And number two, you're locked into a theory that's incorrect. So, and the National Cancer Institute says on their website, cancer is a genetic disease. And everybody mindlessly goes out tracking down genes. We got 1,700 people a day dying from cancer in the United States. That's 70 people an hour dying. And we're throwing billions and billions of dollars at this problem when they have no clue about evolutionary biology and they think it's something other than what it is. And that explains why we're not moving the needle and dropping the death rate from cancer. Not that. Now, that's not, to me, it's maybe for you as well.
Yeah.
The guy on the street is only concerned about how come everybody's dying from cancer. What's going on?
Yeah.
And why are we spending all this money? And then you get good people out there raising money for breast cancer, pink ribbons, all this kind of crap, and nobody knows where the money goes. And we get more and more dead. But if you want cancer to continue killing people, then you support those breast cancer, those pink ribbon campaigns, and you'll get more and more dead people. Because the more and more money we raise for cancer research, the more dead people we get, because they're working on an incorrect theory.
Yeah, that makes sense. Yeah, it's interesting to say that, because you mentioned, I'm not positive about this, but I read your article, I think, and I've researched this. Glycocalyx is so cancer cells have the sophistication and ability to be so sophisticated that, and what the glycocalyx is for our friends listening, is that's the layer around every cell that is basically a signaling system for the cell. So it has just many, many hundreds and thousands and tens of thousands of receptors in this thing called a glycocalyx. And so with what you're saying, cancer can adapt. And I just read an article the other day about they were trying, because cancer cells replicate uncontrollably, proliferate uncontrollably. Well, I read an article stating there was some mechanism they were using to slow down their replication by some kind of enzyme. They were doing it some way because they're trying to adapt to some of the drugs that were being delivered to them. And I don't remember the article I read, Thomas, but it was really fascinating on how cancer cells can adapt.
They don't adapt.
They don't
can't adapt. No. How the Christ are they going to adapt? They have all these mutations in the nucleus. They adapt as long as you have glucose and glutamine in the microenvironment. That's the only way you kill them. How are they going to adapt? They can't do anything.
Well, I don't want to say adapt, but whatever I read, that might be incorrect.
That's based on scientific illiteracy. Say it. It's based on scientific illiteracy.
Okay. That's what I read. Okay. So that's I'm the expert.
Yeah, because these guys don't have any clue about what they're talking about. They don't understand the biochemistry the way we understand the biochemistry. The adaptation of the cancer cell is only because you have not restricted the availability of the two fuels that are driving the dysregulated growth.
So, but if you have those fuels,
yes,
then the cancer cells can make some kind of adaptations on their glycocalyx and their membranes.
No, the glycocalyx is irrelevant. You have to realize all the mutations all that stuff is irrelevant. When you don't have energy, you die.
Yeah.
When you don't have energy,
really
everything is energy.
Okay.
The glycocalyx is synthesized by energy. If you don't have energy, it's meaningless. Okay. You got to realize this. Energy is everything. ATP. And that's what Warburg said, and that's what we say. ATP, energy. You can't build proteins. You can't build lipids. You can't build a glycocalyx. You can't do any of that without energy. You got to have the energy.
Okay,
where does the energy come from?
Fuels.
The energy comes from fermentation. What are the fuels that drive fermentation? Glucose and glutamine. Can they adapt to using a non-glucose glutamine form of energy? Very little. There's two things. You got to have a lot of the fuel logistically. You have to have fuel that can be driving the fermentation machinery. Yes, asparagine can replace glutamine, but how much asparagine is in the microenvironment? Almost nothing.
Okay.
It's not complicated. because we have made cancer an incredibly complicated disease when it's incredibly simple. Because we have run off with these nonsensical people who have no clue about what they're talking about. They're not doing the research like we are. They're not showing the evidence like we are. Okay. There's two problems. Number one, that what the National Cancer says, it's a genetic disease. It's not a genetic disease. Number one, it's a mitochondrial metabolic disease requiring energy. Where does the energy come from? Fermentation. What are the fuels that drive fermentation? Glucose and glutamine. What about fatty acids?
It's that simple. It's that simple.
They can't burn fatty acids. How do you know? Look under the electron microscope and you see them storing the fatty acids in the cytoplasm. Try to get them to burn fatty acids, and guess what? They blow up. The ROS kills them. So we have gone through and we've done all these studies. We published all these studies and we put it out there. And guess what? They don't read it or they don't understand it. So you tell me what the problem is.
Well, and the main, it's interesting to say that because this is really, A plus B equals C and A equals C. This is really very, very common sense or very, very simple. You have two fuels. So for our friends listening, you have two fuels that the mitochondria can use: glucose and glutamine. That's it. The other ones are not available in high enough quantities. Period.
The glucose is coming through the cytoplasm, not the mitochondria. It's coming through the glycolytic pathway in the cytoplasm.
Yeah. And well, the other thing too, what people understand is that the cancer cells need so much fuel that they use the glucose, and they can only make, I believe, from what I've read, there's 4 ATP units in the cytoplasm, and there's 36 in the mitochondrial oxidative phosphorylations. So they need have constant glucose to fuel their processes. So the more sugar you give it by eating cookies and cakes and even bread and whatever it is, the more glucose you give it, the worse off you are.
Yeah,
right.
Well, that's one thing for sure. And the other thing is the glutamine, which is the most abundant amino acid in our body. So it's bathed. Cancer cells are bathed in the two fuels that allow them to. So when you say they adapt, so you throw radiation, chemo, and immunotherapies, and they say, they're still alive, they're adapting. They're not adapting. You just never took away the fuels that they need to live on.
I see.
You want to see them adapt. That would mean if I took away all the glucose and glutamine to the tumor cells.
I see, yeah.
And they were still there, then I would say, they're adapting. But guess what? Nobody has ever simultaneously targeted the two fuels required to manage the disease. And therefore, you don't get the response, the kind that you would expect. So the question is, how come all the top hospitals, oncology centers throughout the world are not simultaneously targeting the glucose and glutamine while transitioning the whole body to fatty acids and ketones? Because that will manage cancer. And the answer is no, they don't know about that. Why? Because they think cancer is a genetic disease. And they're worried about the glycocalyx, and they're worried about all these other things that are largely irrelevant.
Well, and then when you're saying, and this is a really important point, when you're saying that when they say it's a genetic disease, for our friends listening, if that was the case, then if this was a genetic disease, wouldn't 1,000, 10,000 years ago, we'd have the same amount of cancer we have today, or it would be steadily increasing. But in the last 20 years, it's gone up exponentially. Your genes don't change in 20 years. So there's a big problem with that. It doesn't even make sense.
No. You have to separate germline genetic changes, germline, which you're talking about right there, and somatic mutations. Okay. These are collected, acquired from your life living. Okay. So when you say the somatic mutation theory, this is not germline mutations.
Oh, okay.
This is somatic mutations. Like if I take carcinogens into my body, I develop a tumor, and then I look at the tumor and it's full of different kinds of mutations. I didn't inherit those mutations. I acquired those mutations. That's called the somatic mutation theory.
Okay.
And the theory states that the acquisition of these mutations from carcinogens, viral infections, inflammation, all these different kinds of things, those mutations lead to dysregulated cell growth, making cancer a genetic disease. The problem is those mutations are downstream effects of the damage to mitochondria.
Okay.
Therefore, they are largely irrelevant.
I see. So it
then you
technically,
say,
go ahead.
well, why am I doing all these biopsies and having people tell me I have all these somatic mutations in my tumor? And the answer is, I don't know. The information is largely irrelevant because they're not treating you with anything that's, well, we're going to target your epidermal growth factor receptor. Well, only some of the tumor cells have an epidermal growth factor receptor, not all of the tumor cells. Every tumor cell is different from each other if you look at the genetic changes within the tumor cell. However, every single cell within the tumor can't live without glucose and glutamine.
Yeah. But you're saying you're saying the somatic theory is downstream of the problem, which is mitochondrial dysfunction.
Yes.
I got it.
Yes.
That's the key.
Yes.
Everyone listening, now remember that. That's it. Mitochondrial dysfunction. Burn that, right? Tattoo that on your arm. Really. And it's funny, we're talking about that in my book. I have a chart that I make about describing disease and health. And one of the circles is mitochondrial dysfunction and all the factors that cause that.
Yeah.
And then I have another on the other side of the model, the good side, it boosts mitochondrial function and all the factors that cause that.
Yeah.
So just like Thomas is saying, diet, fasting, ketogenic diet, stress management, all these things, your lifestyle factors. That's what people want to hear.
What you're talking about now is prevention.
Yes.
Okay.
Yeah, people want to know.
Okay, listen. You can't. It's very, very, very difficult to get cancer if you maintain good mitochondrial health.
That's it.
Yeah, and we know that from our ancestors who live according to traditional ways, where cancer was rarely ever found.
Yeah.
Well, animals that live in the wild according to traditional ways rarely get cancer.
Yep.
Wolves don't get cancer. Chimpanzees don't get cancer.
Yep.
Very rare, very rare. Domestic dogs are full of cancer.
Yep.
Because they're eating the same crap food that we are, manufactured by dog food companies that don't give a crap about or have no clue about what they're doing. The dogs will eat anything, let's be honest. They're not picky. But they got cancer. We have cancer. Any organism that lives or deviates dramatically from its evolutionary path will have these problems. So, and cancer is just one of many chronic diseases. You can have type 2 diabetes, you can have cardiovascular disease, you can have dementia. All of these are largely caused by mitochondrial dysfunction in one way or another.
Yeah, and that's important you say that, because for our friends listening, it's all these diseases are rooted in the same problem, mitochondrial dysfunction. Just like I write in my book, is that whether it's cancer, diabetes, Alzheimer's, Crohn's, anything, it's all related. It all comes back to inflammation, mitochondrial dysfunction, just like we're talking about. So just like we said a second ago, keep your mitochondrial healthy. Whatever you can do to manage your mitochondria.
And in your book and your chart there, you did touch upon some of the crucial issues to keeping your mitochondrial healthy. Yeah, as you said, you got to reduce stress.
Yep.
You have to do a lot of good exercise, cardiovascular exercises. You have to try to reduce as much as possible highly processed carbohydrates. All of these things will damage and chronically damage mitochondrial function, which could spring upon you as different kinds of chronic disease. Good interpersonal relationships, enjoyment of life, good sleep, all of these kinds of things.
Let's stop really quickly on good sleep, because I read a whole chapter of sleep, and people have no idea the ramifications of poor quality sleep or not enough sleep. It damages mitochondria, it causes inflammation, causes more reactive oxygen species, the circadian rhythms. That's why you need to go to bed and wake up the same time every day, because your body's on a clock.
And neuropsychiatric health, the health of your mind.
Yes.
Depression and all these things. A lot of these things are linked to poor mitochondria.
Yeah.
It's still, that's a very new exploratory area on that, the role of the mitochondria in maintaining mental health.
But it's probably, I'm sure it's correct from everything you've done. It's most likely correct.
Well, Chris Palmer at Harvard is working on that area for mitochondria function in regards to mental health. He's showing very clear examples of where depression and schizophrenia can be linked to abnormal mitochondrial function in various regions of the brain. We are showing the linkage in cancer because cancer, I think, puts the fear of God in so many people, and then when they are found to have a tumor, the kinds of treatments that the medical establishment uses also puts the fear of God into people because they don't want to be poisoned and irradiated or surgically mutilated. And this, when you hear that term, and then you go to the oncologist and we immediately have to give you chemo or radiation or chop out some part of your body, that gets people very, very upset. And metabolic therapy shrinks the tumor down significantly and then leads to a potential cure from surgical removal or even a radiation now that the tumor is localized in a specific area because you've deprived it of its two fuels. It shrinks, becomes small, becomes very manageable now, not unmanageable. So I think in the long term, we will be able to take away a lot of anxiety for people. We can use chemo, but we use it in very, very low dosages. So you don't have to have hair loss and you don't have bleeding gums and people are always concerned about your microbiome, microbiome this, microbiome that. And then they go out and get irradiated and poisoned. It blows the hell out of your microbiome, when we learn that cancer is a metabolic disease, mitochondrial metabolic disease, we will be able to manage this disorder with far more success and far less toxic adverse effects.
Now, when you were saying about the chemo on the low dose, for our friends listening, chemo therapy is, from what I understand, they use plants. So plant toxins, I can't remember the solanine and some of these other things that are, they use those, I think sulforaphane, but in very high quantities. So it's basically can affect the cancer cells and the proliferation of the cancer cells and the way they they're functioning, but it also damages healthy cells. And so when you're saying about low dose, Thomas, is that low dose will damage fewer healthy cells than if you kind of just bomb everything. Is that correct?
Yes, absolutely. So I mean, why are we poisoning somebody to kill cancer? Well, they're dividing out of control. Cancer is cell division out of control. So if we use a chemical that blocks DNA replication, we kill the tumor cell, but we go bald and we blow the hell out of our gut. And all the other parts of this normal cells, like you said, that need to grow normally are now harmed by saturation chemicals throughout the body. Or we incinerate, we radiates the side of some guy's head or his throat or some part of his lung or whatever. And we damage a lot of the surrounding tissues doing that. Some cancer cells, they need a cytoskeleton. So you use these vincristine and these horrible drugs that block the cytoskeleton so the cell can't divide. But listen, the cell can't divide without energy. So where is the energy coming
to that? Absolutely.
Where the hell's the energy coming to allow the cell to divide in the first place?
Yep.
And it's coming from glucose and glutamine. So we can target those two fuels without the toxicity. So why don't we do that? I don't know.
Well, it's when you're saying that, a fourth grader would understand that. Really? I mean, it comes from glucose and glutamine. If there's no fuel, there's no cancer growth.
Yeah, but now you have to be
not
careful.
Hard.
Yeah, well, you have to be careful on how you target the glutamine. Glucose is easy, but there's again, when you tell people, well, what do I have to do? Zero carb diet, water only. Well, I don't want to do that. Okay, then go take the radiation and chemo. It's your choice. It's just like, okay, we're not telling anybody, listen, I'm not standing here to deprive anybody if they want to get their body nuked or they want to take a poison. That's their personal choice. But I know there's I would never do that.
Give them information. You're giving them information that they can use or not use.
And the problem now is if you go to the oncologist, they never heard anything that I've just told you. They have no clue. Never heard of it.
Well, it's
So
getting better now, isn't it? More people are starting to learn about this.
lay people are, not the establishment. The establishment still remains massively ignorant of the whole fact. But yeah. So anyway, when you target glutamine, you have to be very, very careful, because that same fuel that drives the dysregulated growth of the tumor cell is also extremely important for the health of our immune cells and our gut and our urea cycle. So you can't go after glutamine like a pit bull on a hamburger. You have to be very strategic in how you target the glutamine, and that's why we know there are drugs that we use pulsing. That's why we developed the press pulse therapeutic strategy, which will eventually become the standard of care once people realize what the hell I'm talking about. And once
Well,
they
now
understand.
let me ask you a question. I was watching a video from a gentleman, oh God, what's his name? Casey Pulliver or something. He mentioned you. He talks about the mitochondria in depth. And I've watched a lot of his videos. He says, and I'm not positive about this, but this is I wrote down. Vitamin D helps to inhibit the glutamine pathway in cancer cells. Is that correct? Or is partially
No,
correct?
I don't know about that. We need vitamin D. It's an important. It's a very important vitamin. But we don't forget. Why would we want to do that when we can just target the glucose and the glutamine? I don't know.
No, he's saying the vitamin D, when you have sufficient vitamin D, it helps to inhibit the glutamine pathway that the cancer cells use.
Oh, well, that's
Is that correct?
no, vitamin D balance is healthy for the body. You need it. So you're talking about prevention. But once you have developing cancer, you have a different, now you're going after the fuel sources. We use the drug deoxynorlucine, Don, which unfortunately, that to me, that should be the drug that everybody who has cancer is being given, and it's not given to anybody at all. It should be the number one chemo drug should be Don. Deoxynorlucine when administered together while the patient is in nutritional ketosis. We would drop cancer death rates or keep people alive with a higher quality of life far longer than we're currently doing.
Is that
So
the
the
press
question
pulse
is,
therapy? Is that the press pulse you're
yeah,
talking about?
but you press glucose down because you don't need glucose. You can substitute ketones for glucose. The brain will switch to ketone bodies rather than glucose once you press it down. We evolved to do that. We have to take advantage of our evolutionary capabilities. We have to know how our cells work, so we can marginalize the tumor cells, and then we have to strategically go after the glutamine gently and strategically. You can't just go and press it. You pulse that.
No, makes
So
sense. Okay.
and then when the body is in nutritional ketosis. So how do you know that? Use the glucose ketone index meter that I developed. The calculator that our lab here developed, so cancer patients can know when they're in have a low GKI, and then they would take the Don and elevate their ketones, the Don drug, the glutamine targeting drugs, and a knowledgeable physician would be able to administer this therapeutic strategy to the cancer patients, and have the cancer patient together with the knowledgeable oncologist or physician. They would be able to work together as a team to gradually reduce the tumor growth without collateral toxicity. The problem is neither the patient nor the oncologist know anything about what I just said.
Yeah.
Even though the papers are written in the literature, even though they're there, and even though the outlines are there, they don't know that because they were never trained in medical school to know that. And the National Cancer Institute says it's a genetic disease, so why should I know about glucose?
Why they bother? So let me ask you another question that I found really interesting. The fulvic and humic acid. I write that on my model how important it is. It's a metabolite made by the soil that's not in our food now because our soil has been so depleted and damaged. I read a paper probably 3 or 4 years ago stating that fulvic and humic acid can help attenuate cancer cell metastasis by not allowing them to attach to the endothelium. The paper was fulvic acid attenuates colorectal cancer cell, resistant-induced colorectal cancer cell adhesion to the endothelium. I thought that was fascinating. You know anything about that?
No, I don't worry about that. So, yeah, I know how to kill cancer cells without toxicity.
That's
I
all that
don't worry
matters.
about all that stuff.
All the other stuff.
I'll tell you what, once we get rid of the guy's tumor and he's healthy, we can go back and start
Take a
looking
fulvic
at
acid.
those other kinds of things, right?
Yeah.
So I know what metastatic cancer is. It's actually a fused hybrid between our immune cell and some sort of a stem cell. And then when I looked at this, we spent decades looking at this. And then I found it took me 10 years to figure out what the biology of the metastatic cancer cell is. And it's a macrophage.
Yeah,
And
you said that in one of your
corrupted
articles. Yeah.
macrophage. A macrophage is one of the most powerful cells in our body. It's like your best friend. What's it called? The Marine Corps. Marine Corps says, "I'll either be your best friend or your worst enemy." And the metastatic cancer cell is a macrophage. It already can move around the body. It just becomes corrupted. So then you say, okay, it's a macrophage. Then we jump in and what is about tell me all about macrophage biology. Okay. What do they eat? They're heavily dependent on glutamine. Now we know how to kill them. And then you have to do it so strategically so you don't harm the normal macrophages in your body.
So are you saying that the this is interesting. Are you saying that the cancer cell attaches onto the macrophage and most of the bloodstream, or it becomes part of it?
Yes, it becomes part of it.
Wow.
So what happens is the cancer cells they're usually epithelial cells. They have no capacity to metastasize or invade. They don't have you have to have a certain biology that allows the cell to move around the bloodstream and enter and exit tissues. So the macrophage already has the programming to do that. It has the genetic architecture to do that.
Yeah.
But what happens, the macrophage is a wound healing cell. Yeah. So when you have a group of cells in a particular organ, in a particular tissue, and they're growing out of control, they create a disturbance in the microenvironment that signals our immune system to come in and say, what the hell is going on here? It's like if I had a cut, the immune system would come in, kill bacteria, and the fibroblasts and the macrophages working together would form a scar, and you would heal the wound. Okay. But cancer cells are growing, throwing out the signals that make them look like a wound. So the macrophages come in as a wound healing cell, and they throw growth factors and cytokines that would normally facilitate wound healing, but it's like throwing gasoline on a fire. It's making the tumor cells grow faster. And in order to facilitate wound healing, they actually fuse with each other and with other cells to try to facilitate wound healing. Wow. So now you have this hybrid cell of a neoplastic stem cell fusing together with our normal immune cell, corrupting now the macrophage itself. Wow. So it becomes a rogue macrophage, and it has biological characteristics of a macrophage, which can enter and exit the bloodstream and go into tissues, called metastatic cancer. Wow. It's a rogue macrophage. So how do you kill it? Well, it's eating glutamine and glucose. I mean, it's just...
that's fascinating. So basically, it's like the cancer cell, for our friends listening, is like, okay, we hijacked a bus, basically, to get us into the bloodstream.
Yeah, but it's not a conscious. It's not.
Well, no, I know, but it's kind of like that's an analogy. So it's able to use it to go into the bloodstream.
Yeah, well, that's we call them rogue. They're rogue macrophages. Metastatic cells are rogue macrophages. Wow. And they go to certain tissues. They love to go to the liver and the lung, and these kinds of things. So there was a study done some years ago where they took isolated macrophages from one mouse, labeled them, and injected them back into another mouse to see where they would go. And they go to the lung and the liver.
Wow.
So why? Because the lung, when we're breathing air, we're bringing in a lot of particulate matter from the outside. In fact, lung cancer is still the number one cancer because even when people stop smoking, we drop lung cancer for sure and other cancers, but lung cancer is still the top number one killer of cancers because we're always breathing particles from the air. Okay, so that damages cells in the lung tissue, and the macrophages, we have a lot of what we call resident macrophages in the lung that are designed to gobble up these particulate matter. And eventually some of these particulate matter corrupt the macrophages in the lung, leading them to become metastatic. They already have the capacity to do this. And if some of the other cells are damaged, the macrophages in the lung will fuse with them, and then you get this spreading all over your body. And that's why lung cancer is so deadly. It spreads very, very rapidly because the lungs are loaded with macrophages to protect us against the particles that we breathe.
So I understand this. the reason, because if the cancer cells were floating in the blood by themselves, they would be targeted by they'd be killed because they'd be targeted by macrophages to basically as a foreign invader. But when they become part of the macrophage, is it kind of like it's kind of incognito when it's going through the blood vessel, so the other macrophages don't, get the cancer cell and kill it before it gets to go somewhere in another tissue? Does that?
Yes.
Oh, that's fascinating.
They're recognized as self.
Wow! Wow! For our friends, that's fascinating. Wow! Wow! I'm sorry, I'm blown away. So they're incognito.
Yeah, they're saying, why am I going to kill this guy? He's one of us.
Wow! Wow! That's mind blowing.
And so you have a big mixture of things, and some cancer cells will be killed by macrophages that don't have their identity markers.
Wow.
And some won't be. So it's kind of a hodgepodge. But that's why they always say, well, how is it possible that cancer cells can pass through the bloodstream and not be killed by our immune system? Because they are the immune system.
Wow.
They are part of our immune system. So they're being pat on the back as they're going all over the place.
They're cloaked. They're basically cloaked and hidden. So
they look like I have all the identification of being one of you guys. So they don't recognize it as being foreign at all.
Wow.
So
fascinating.
And when you say immunotherapies, they're supposed to target and kill some of these cells. The problem is you have normal cells that have the same markers as some of the cancer cells. So you have this problem with immunotherapies called hyperprogressive disease. The drug companies aren't hiding any of this stuff. If you listen on television at night to the Opdivo and Keytruda commercials about these cancer drugs, immunotherapy, they spend more time telling you how the drug will kill you than how it's going to kill. But they're not lying.
No,
it's true.
Wow.
The damn drugs will kill you sometimes faster than the tumor will. So it's not. But why? I said, why do that when they can't grow without glucose and glutamine? So why are we using those stupid things in the first place?
It's all irrelevant.
Yeah.
Without targeting glucose and glutamine.
If you have a tumor and you've restricted glucose and glutamine and you've kind of trapped them, now you still have something that's there growing very, very slowly and seems to be able to survive on small amounts of something else in the microenvironment. It's still not growing aggressively. It's just kind of hanging on. There might be some commonality that all of those cells have together, allowing them to still be alive after you took away their fuels. Now, an immunotherapy that is specifically targeting something in common will kill them all and get rid of it. So the immunotherapy should be like after you go to the dentist, he's putting fillings in and doing all this stuff. The end of your stuff is the teeth cleaning. So you come in with an immunotherapy at the end and you possibly could eliminate whatever residual cancer you might have had left. It's a stage.
So you're saying do it at the last stage is the
do it at the last stage. Sure.
Yeah.
The same thing with radiation and chemo. If you put the patient who has a tumor in metabolic therapy and you shrink the hell out of the tumor, but you don't get rid of it all, but you know where it is, now you may be able to surgically debulk it, or you may be able to nuke it, or you may be able to poison it with minimal damage to the collateral area, because these guys are just barely hanging on because you've starved them and they're just like residual. They're there. You just got to push them a little bit over the edge. That's where you can use these other therapies that we've been using as an upfront approach. They can be used as the final cleansing of the body of these things, but you do it strategically in low dosage, timing, scheduling, and that's what we're working on. And that's our research right now. And we get our funding from philanthropy and private foundations. So we have a people who want to be part of this paradigm change, the moving cancer from a deadly kind of a dismal situation to something. Well, it's never going to be easy, but I can manage it and I can live on, I can get a higher quality of life. I can live longer. I don't have to lose my hair and have my microbiome blown to hell. So as long as we can keep our preclinical systems working, which have direct translational benefits to the clinic, and we'll set up clinics that will, based on our research, we will be able to manage cancer in the majority of people, especially when they're first diagnosed.
Yeah, and I think that's important. I think too is that, like we talked about earlier in the conversation, and that this is a lifestyle problem, and it's not it's an insufficiency of chemotherapy drug problem. It's a lifestyle problem. and that
Well,
we
we
have
know
to work on that. Yeah.
we know the vulnerability of the tumor cells. We
Yeah.
know what they need to survive, and we know how to target that. And then once we know that, we'll be able to manage this disease far more effectively.
But with what you're doing, the great work you're doing and your colleagues showing them this and getting this message out there is super beneficial because, like I said, I've had family members pass away from cancer. It was horrible. It's
Yeah,
we all have somebody know somebody,
yeah.
and
Well, because
it's horrible.
yeah, we haven't been treating it the right way, and we've been ignoring the signs of the cancer telling us what we need to do to kill them, and
Yeah,
we haven't been listening to the signs.
yeah,
So now
yeah.
we are.
That's good.
Now, how long will it take for the government to know this? I don't know.
Well, the great work you're doing. Um, anyway, I'm gonna let you go. I really appreciate your time, Thomas. It's been fascinating conversation, and very grateful for what you and your team are doing at Boston College. There, keep doing that work. How can people find you? Where can they find you to learn more about your research and all that?
Well, most of my work is open access publication on the web. I just go into look at my name, say what do I publish, and people can download the breast pulse therapy. They can download all of these things that I've been talking about. They're open access, which gives everything that and so much more. I could go for days talking about this stuff. They but you're talking about a lifetime's worth of work here, and it's in publications that are on the internet. So people can go and look at all this. All the key things that I've been mentioning are open access. And the big question that people say was, how come the field isn't looking at it? Because they think it's a genetic disease, and when
Going back,
it's
yeah.
called consensus um, uh, confirmation bias and dogmatic ideology. It's a very powerful effect on the human brain. I mean, you know how powerful religion is. Some people would rather die than change their religion. Why? Because it's indoctrination of the brain into that particular viewpoint. And science is very similar. Once you're indoctrinated into a particular scientific perspective, you can present all the data in the world. You're not going to make a change. So it has to come from successes after success after success. And,
Grassroots.
and people say, well, how come there's no clinical trials to support what I'm saying? Well, who's going to do a damn clinical trial on this? When you think cancer is a genetic disease and you tell them it's not, well, then I don't want to do a clinical trial on it. I mean, it's like everything makes sense until you get enough people saying, I want metabolic therapy, give me metabolic therapy. Where is the outcry in the population? They go like sheep to these oncology centers. And then if they do survive, and many of them do, we have millions of cancer survivors throughout the world, they pay a significant price. Their bodies are beat to hell in one way or another. Many of them, not all of them. But they have all kinds of other health issues that reduce their quality of life. We shouldn't do that. We should have a higher quality of life after surviving cancer using metabolic therapy. So,
Absolutely.
yeah. And as long as I said, and when people support our research, we acknowledge them in our acknowledgement section of the papers.
Good.
So if people read my papers and they go to the acknowledgement section, you see all these names. Who the hell are these people? Well, they're the ones who gave us the money to do the research.
Good. And that's fantastic.
So if people want to
Yeah.
become famous, what they do is they support our research. I put their name in the acknowledgments, and they can show all their friends how they've contributed to the paradigm shift.
Well, and like I said, you're doing fantastic work,
And we have
we
the
appreciate
money.
it.
The money goes directly to Boston College to support our research, our Cancer Metabolic Fund, or Travis Christofferson's Foundation for Cancer Metabolic Therapy. Those are
Right.
the supporting groups that we get. Yeah, so people get excited about this, and they should be, because this is going to be the future.
Yeah, it's fantastic.
It's just that it needs to get out there, and guys like you are going to help do that.
Well, thank you so much for your time, Thomas. I really appreciate it. It was just a fascinating conversation. And I don't know what else to say. It was a pleasure talking with you. It was an honor, and keep doing the good work. And thank you so much.
Well, thank you.